All right. Well, sorry, I was just fixing the lighting conditions in here. Can everyone hear me and see me okay? Check the chat here. Yes, okay, awesome, great. Well, welcome back, everyone. This is the second to last edition of this course. Today, we're going to talk about finishing up. We're going to finish up processing the single cell data that we started yesterday, and then we'll talk about visualization and getting like gene lists. So should be a good day. All right. Just a reminder, everyone, you have questions, put them in the chat, put them in the issues section of the GitHub website for the course, and we'll get to those whenever we can. If not during the talk today, we'll get to them afterward. Okay, so when we chatted last time, we sort of left off here in the load process script. I want to just point out to you that this script is available to you. Basically, something very similar to this. I'll point out some differences, but if you run this command in your R session and you have the Blazor templates package up to date and loaded on your computer, you should be able to load this exact script into whatever file that you want. So that's easy to get, and then you just have to modify it in a few places to perform the way you want it to perform. Okay, so last time we stopped after, right around here where we were predicting doublets in our data. So that's done with this command. It's a map command, and it's using the doublet finder function from the Blazor tools package. That particular function is one of the longer functions. Maybe takes for this on this computer takes 4 or 5 minutes to run. So if you're running a large data set with 100,000 cells, that might take overnight to run. So that's just something to be aware of. That predicts sort of synthetic doublets, or how your data lines up with synthetic doublets that it creates from the data, and it marks those, and we'll get to that in a minute. So after you've gotten the quality control steps done, that's the QC per cell, looking at mitochondrial gene percentage and the doublet finder, the next thing you have to do is rejoin those data back into a list, okay? And that's just run with this command here. Here's a question: did you write these functions like doublet finder or import them from Seurat? So everything that is prefixed with the BB, that just tells you that I wrote that, and it comes from either the Blazor tools or the Blazor templates package. In many cases, it incorporates Seurat functions, or in some cases, it incorporates Seurat functions. I do that to sort of abstract away things that are repetitive or like hard to look at or easily messed up that you don't want to, ever change. And so we have a lot of those functions that sort of wrap one or two or three or more other functions from other packages. So that's sort of how these functions came to be. Okay, so once you have a list of your CDS objects with the quality control information in them, the next step you're going to do is you want to take that list and put it into a single CDS object. All right, and this is a function straight out of Monaco 3. It's called combine CDS. It's very easy to run. It's very fast. All it does is it joins everything together. It takes the matrix, the gene expression barcode matrix from each of those CDS objects and just basically binds them together side by side. It generates unique names for your cells in the process of doing that. So one difference to point out here from the way you're normally going to run this is that I've named each of these objects that are being created in serial fashion. So CDS main _1, 2, 3, and it goes down to like 14 in this particular script. Reason for that is just for the purposes of this lecture so that we can save those individual objects and explore them, here with you. When you're running this yourself, you don't have to save all these objects, and you definitely shouldn't save them. You can maintain them serially in memory, like I'm showing over here, all these CDS objects, if you really want to investigate what's going on. But usually what I will do is I'll just name these all CDS main, and then each step overwrite the previous step. And that just saves, on memory consumption for your computer. So that could be something to make it more efficient for you. In any event, there's a few steps here that are pretty quick to do. You combine all the CDSs together, you remove, and you can omit this step if you prefer, but I usually will remove the mitochondrial and ribosomal genes, which are, basically uninformative for the most part for what you're going to do downstream from here. You're going to remove the cells that have been flagged as low quality because you definitely don't want them. And that brings us to this section here. Okay. Now, if you read this comment, it says, uncomment and run the following section if you want to preview the CDS before removing predicted doublets and aligning. Okay. So there's a lot in there, but basically you have some decisions that you're going to make as you're analyzing these data. And one is, do you want to remove doublets? And if so, do you want to remove the high confidence doublets or the low confidence doublets? You could just remove the high confidence doublets and not really worry about it. So that's definitely a possibility. In which case you could just leave this all commented and just skip it. But in this case, I've run it just so I can sort of demonstrate for you, what the doublets look like. And so I'll show that very briefly now. So basically, if we look at CDS main 5, we can plot the predicted doublets here. And I'll just zoom in on this for a second here. And you can see that, the doublets are highlighted here in this orange color. And, there's no like distinct cluster of doublets. And, there's not that many of them. There's like 8 for the whole data set. So I really don't think that there's a major problem with doublets in this particular data set. So you could either like totally ignore it, or you could just remove those 8 cells and it's really not going to affect your data that much. So that's, now if you're running like say, 10,000 cells per sample and you're going to have a doublet rate that might be, up to maybe 10%, this is going to be more important for you to do. In any event, that's what was done here. But we can choose, and I usually choose just to remove the high confidence doublets, and that's done with this command here. So what happens is now you've got a subset of your CDS that was, in the dimensions, the UMAP dimensions, which we're going to go into in a little bit more detail in a moment, the UMAP dimensions were, calculated based on the full data set. So you want to generally, if you're going to do something like that, you want to recalculate the PCAs, and you're going to want to recalculate the UMAP dimensions. Okay, and so that's more or less what's done in these next few steps. First of all, I remove those columns from the cell metadata, and I'll just show you how that looks. So, if we go to this guy here, we can look at the cell metadata for, let's say, CDS main 5. And I'm just going to type in this command here, glimpse. All that does is it shows us all the columns that are in the CDS cell metadata. Okay, and you can see here you have doublet finder low confidence, doublet finder high confidence, and the QC any. And so what happens when you get to 7 here is you're going to remove those guys. Okay, so if we can look at 7, and we're just removing those because we don't need them anymore. They're not helpful for us. And you want to keep the size of your data objects as small as possible. Okay. So the next thing you're going to do is you're going to calculate the PCA dimensions. Now, I'm not going to go into like a mathematical discussion of what PCAs and UMAPs are. Basically, they're just ways of going from 30,000 features for a particular cell down to a smaller number for PCAs, and usually that number is between 30 and 50 or 100, something like that, and then condensing those 30 or 50 PCA dimensions down to 2 dimensions for UMAP. And the reason it's done in that way is there's different calculations that are performed downstream that use either the PCA values or the UMAP values. So, the CDS object is designed to hold those data, sort of hidden away in like slots. Okay. And, you usually will not be directly interacting with those particular slots in your data, but it's useful to know that they are there. And I'll show you for just 1 second, where those are at. So if you run this command, single cell experiment, and then the command is reduced DIMS, and you run that on the CDS that you've made, that will bring up an object, okay, a list, and that has all of your UMAP dimensions in it, all of your reduced dimensions in it. And you can actually look at them like so. And you can see here that each cell now has has 2 dimensions. Okay, and those are just 2 numbers that describe it. If you run the PCAs, you get more. Okay, so there's up to like 50 principal components for each cell. So that's how the dimensionality reduction is done and is stored, in this workflow. Okay, so you've removed the low quality cells, you've calculated the reduced dimensions, and now basically, you're almost ready to go. The last question that you're gonna ask yourself is, do I want to perform any alignment? And alignment is sort of a fluid step in this. So you can, align by 0 factors or 1 or more factors, basically. And the reason to do this is to it doesn't, so the thing is, it doesn't change the underlying expression data for your cells. What it does do is it changes the dimensions. It actually calculates new principal components and then calculates new UMAP dimensions based on those principal components. And the purpose of this is to change the visualization and the clustering of cells. And so this can be very useful if you have a very heterogeneous data set that you would like to reduce the heterogeneity to help streamline your analysis. So if you have, let's say, a sample of cancer cells from, even multiple patients with the same type of cancer, they're all going to be very different. And you can use alignment based on the sample factor or the patient factor in order to bring all of those cancer cells together in the same space and identify them more easily. So, but you can choose a lot of different factors from your analysis to do that. Whether it's the sample or it's batch or some other technique or time point, you can choose any one of those things. So that's something that I can't really prescribe for you, but is going to be something you're going to want to explore probably. Okay, yeah, so then basically what happens with this particular function, BB align, is what it does, is it does the alignment using a package called Bachelor, and then it recalculates the PCAs based on the aligned values, and then it recalculates the UMAP coordinates based on the aligned PCA values. Okay, there's a question: how does the program go from all the PCA dimensions to just 2 dimensions? That's the UMAP algorithm. It's sort of a, I can't really explain it, because I don't know the mathematics in great detail, but more or less the idea is that it plots, it projects the 50 dimensions, the 50 PCA dimensions for each cell into a 2-dimensional manifold, from a 50-dimensional manifold to a 2-dimensional manifold, which is basically a plane. So that's just a command that you run, and you don't really have to know in detail how it works. The important things to know that I am familiar with and that you need to be familiar with is that every time you run it, it's going to generate slightly different results. And they're not different in a important way. They're different in trivial ways, but it makes it not reproducible. So, if you're going to sit down to work on your project, you're going to want to not want to rerun that step every time you sit down to work on your project. You're going to want to save it, and you want to ideally save it into a data package, which is what we're going to talk about next week. So that's a good question. There's a sort of a random seed involved, and if your computer's using multiple processors, it's just not reproducible. So so you only want to run the alignment or the dimension reduction samples commands once. Okay. Another question: when doing alignment, what is the program aligning the data to? Is there a reference genome? This is not aligning to a reference genome. We'll get that later. It's aligning based on factors. Okay, and in this case, what I've told it is the factors in your own data. In this case, I told it to align it by sample. So if you look, we can just run, CDS main 9, and then look at that. It's aligning based on this column here, sample, and so it's going to make, all of the cells, that are from the Chromium controller, and all the cells that are from the Chromium X is going to make them line up more or less to their nearest neighbors. It uses a K nearest neighbors algorithm to do that. So it's just inherent based on your own data. And you could have picked any one of these. You could have lined it by equipment or date, or whatever. So you just have to play around with that. All right. So the next thing that we're going to talk about is clustering. Clustering is a critical step for what you're doing. That's how you more or less identify cells that are similar to each other. You're going to do a lot of your calculations and your analysis based on these clusters. And the important thing is that these clusters are generated in an unsupervised way. They're generated algorithmically, and so they can be relied upon to identify, groups of cells that are in a biologically similar transcriptional state, and that's the important thing. Now, there's many different algorithms that you can use to do this. I've written this function here called triple cluster, which will cluster the cells based on 3 different algorithms. One is called Louvain, one is called Leiden, and the third one is called partitions. Okay, and the way I think about it is coarse, medium, and fine. So partitions are the coarse ones, coarse clustering. Louvain is the medium, sorry, Leiden is the medium, and Louvain is the fine clustering. You run this command, and what this will do is it will identify those clusters, and it will print out the top specific genes, however many that you want. Usually I just do 50 for each of those clusters and save that to a CSV file. So with this one command, you cluster your cells and you get all the top markers from those cells. And so let me just show you a little bit of that. So this is from a different version of the CDS, but it demonstrates what we're talking about, okay? So if you look here, you can see the coarsest clustering is the partitions, and really there's only one partition. So all these cells are blood cells, and it couldn't, using this particular algorithm, it couldn't identify any differences. Leiden is the medium clustering, and that actually seems to be a pretty useful way to cluster. Those cells are, pretty logically, in different groups, and so that's good. And then if you really wanted to drill down into transcriptional states that are more specific, then you can use the Louvain clusters. And so you have a lot of things here that you can choose from as well. All right, and so you're going to have to decide based on your data and how heterogeneous it is and what's going on, you have to decide which one of those clustering algorithms you want to use. You can bin clusters. So let's say you wanted to bin like these 3 clusters and these 2 and these 2, and then use those as new clusters. That's something that you can do pretty straightforward in a pretty straightforward way. Another important thing, like the UMAP calculations, these are non-deterministic calculations. They have a random component to them. And so if you were to run this again on the same data, it's going to come up with slightly different results. So this is another thing that you want to run once and then save the results. Okay. So when doing clustering, can you specify how many clusters you want? I thought in Seurat there is a command you can specify resolution. There is. And if you want to do that, you would have to use, in this case, the monocle function to do that, which is, I believe, cluster cells. You can look at the monocle website to do that. I don't find that, and this is the reason why I haven't incorporated it into the triple cluster functions. I don't find that to be a particularly useful thing to do. That when you're getting into like changing the fine parameters, your clustering method only gives you coarse, intermediate, and fine, but you can't specify. No, you can. What I'm saying is that you can change it if you use the internal function from Monocle. You'd have to use that one from the Monocle website. Yeah, I just don't find that to be a particularly useful procedure to do, which is why I didn't incorporate it into my function. We can talk more about that later if you have any other questions. All right, so let's go back to this script here. We've generated now the cluster information for the CDS, and we can just look, for example, at this, and you can see that, actually, what is it here, 11? It's 12, we'll look at 12. So if we look at CDS main 12, we can see that the Leiden, partition, and Louvain clusters have been identified and added to the cell metadata. So that's good. Next thing to do is to identify gene modules. Okay, so gene modules are a particularly powerful way of identifying groups of genes that or gene signatures that are inherent in the data. So you could say a module is like any set of genes, but when we're doing it here, and this specifically uses a monocle function, which is one of the reasons why I like monocle, is that it identifies in an unsupervised way groups of genes that are co-regulated from cell to cell in your data. This command will identify those modules and then add them to the row metadata for your CDS. So if we look at not the cell metadata, but the row metadata for CDS main 13, you can see that what's been added here is columns for module and supermodule, okay? And a module is like, might be anywhere from a dozen to like 1,000 genes or 2,000 genes. And a supermodule is, maybe, a group of modules, more or less. There's no real difference between module and module label. It's just the way that the way that factor is printed. In any event, these are important things that you're going to want to run. So I always run that on all the data sets. And basically the last thing that we do is also an optional thing, and that is aligning to a reference data set. And people get really hung up on this, and they like spend a lot of time like identifying cells. And I think the important thing is that this is like a garbage in, garbage out type situation. You're not always going to be running something that can be aligned to a high quality data set. Now, if you're running human PBMC, then maybe you can do that because there's a good reference data set provided by the Seurat people that will do that. And in this case, this is what I've done, is I've aligned to that reference data set. But you may not be running human PBMC, you may be running like a cancer sample. And if you were to align a cancer sample to the Seurat data set, which is normal human PBMC, then, like a leukemia cancer sample, then, it's going to identify leukemia cells as monocytes or dendritic cells or something like that. That's not what they are. They're leukemia cells. And so you're going to have to take anything and evaluate what's the label that's been applied and make a judgment, and then support that judgment with data and visualization and other things in your project. So this is something that I ran on this particular data set because it happened to be human PBMC, so it lines up well and gives you a good set of labels. But this is definitely not something that you should be running on esoteric data sets. And you can't run it. For example, if you're running a mouse sample, you can't run this particular command because there's no mouse, like say bone marrow data set provided by the Seurat people. So you have to use a different alignment procedure, and that gets to be a pretty technical thing to do. So, so I think that's definitely an optional thing. Now, if you are running, PBMC samples, this is definitely something that you can try. You do need to have this particular file to make it work. If you ask me, I can tell you how to get this file. It's actually available from the Seurat website, and you have to provide it. The way I've done it here is with this command, but you can just put the path to that file on your computer in there. And yeah, so that's a particularly useful thing when the reference is good for your data. Okay, so let me show you. So basically that's sort of the end of the scripted analysis that I usually run, and then I will save all these objects like you see here. And this is now saved on the hard disk, and that will stay, saved. It's not in memory. It's saved and written to the disk. So that's good. Now, let me show you what the next thing is, and this will take us to the end of what we were supposed to cover last week, which was for sure a lot of stuff, and that's cell assignments. Okay, and so once you've done all this stuff, this is pretty much can be more or less automated, and you can just click this source button and it will run, and then like it should all run to completion. But then you're going to want to start to look at your data and say, okay, well, these cells are T cells, these are B cells, these are whatever they are, and then that's like a supervised assessment that you're making based on data. And number one, you can add that and save it permanently in your data, but you can also go back and change it. And so the way I usually do this is I'll run this, save the objects, and then every time I go back, and say, okay, well, I want to rename these cells based on X, Y, or Z things that I found, then you can do that and then resave it. You can also not necessarily resave it if you don't want to. The renaming of cells is a very fast, lightweight operation, which I'll show you in 1 second. Okay. All right. So, question. Did you want to look at differential expression between cluster A and cluster B, or cluster A versus all other clusters? How would you do that? All right, we're going to get to the end of that. We're going to get to that at the end of the day today, hopefully. And if not, we'll cover it next week. All right. So let me show you how I do the cell assignments. Okay. So the first thing that I do is, we've already sort of explored, what these clusters are here. And, for today's demonstration, we're going to look at the Leiden clusters, because these look like, it's like a manageable number of clusters that we can identify. And it seems like it might be useful to name those something other than 1, 2, 3, and 4. Okay. So what I'm doing here is I'm going to run this command, which extracts the cell metadata. It's going to group that cell metadata by the Leiden cluster, so 1, 2, 3, 4, and then it's going to provide the it's also going to group by the Seurat cell type that was generated. This is the automatic name that was given to those cells by Seurat. And it's going to summarize that table to provide just the count of each of those combinations. Okay, so I'll run that very fast. And that gives us this table here. It's very, very concise. So Leiden cluster 1 looks like it's got a lot of T cells in it and some NK cells. Cluster 2 is mostly monocytes. Cluster 3 is mostly B cells. And cluster 4 is mostly dendritic cells, at least according to the Seurat automatic classification. Okay, so what you can do is you can just plot that, and just to give you a visual idea of what's going on. And it's very clear that Leiden cluster 1 is pink T cells, NK cells, CD8 T cells, CD4 T cells. Those are all T cells. 2 is monocytes, so they're all monocytes. 3 is red, that's all B cells, and 4 is almost all dendritic cells. So that's a pretty clear breakdown of what those cell types are. And then from there, it's very simple to rename those clusters. And you don't want to overwrite the Leiden cluster. What you're going to do is make a new column called Leiden assignment. Okay, that's the way I always name it, but just name it something that doesn't overwrite the original cluster information. Okay. And so that's what that does there. And that's you can see how fast that ran. So that's something you can do sort of like every time when you're running your script. That doesn't break anything in your data. So that's a good one to do. And then we can now plot those like so. And now you can see that these clusters have all been named. So TNK, monocyte, B cell, and dendritic cell. And then you can also look at the cell metadata. Okay. And you can see that those have been added down here as a new column, Leiden assignment. Okay, so that's pretty much what I do. I don't always, as I said, use the Seurat automated labels. Most of the time you're going to look at the gene lists that were produced when you did the clustering, look at the top specific genes, and then make some judgments based on what those things are. But you want to incorporate all that information and use your scientific judgment to sort of figure out what these cells are to give it a name that's useful for your project. Okay, so that is pretty much the end of what we wanted to get through last week. And so this week we're going to talk about visualization and differential gene expression. So I'm going to go over to my notes here. Now these notes are available on the website for you guys. So I just wanted to think about and discuss for a moment the goals of what you're trying to accomplish with good visualization for single cell data, because there's definitely principles that you want to adhere to. The goal is, number one, in my mind, to demonstrate global transcriptional state in single cells. So that's what your UMAP dimensions are. That's the global transcriptional state of a cell broken down into two numbers. You want to demonstrate groups of cells that are in a similar transcriptional state, and we call those clusters, and I just showed you that you can use small, medium, and large clusters to do that. And as was discussed, you can even fine-tune that by using some of the monocle functions if you want, but usually I prefer to let them run and identify themselves without too much interaction. You can, you might want to show the distribution of sample level metadata according to transcriptional state. So, all the things that you knew about the samples before you did the experiment, like what mouse it came from, what patient, what was the immunophenotype, the genetics, whatever. That might correlate with transcriptional state, and you might be able to show that in your data. You may want to show expression of single genes or groups of genes in arbitrary groups of cells. So, going in, you may not have realized that there were one or more, sort of unanticipated transcriptional states for your cancer cells, for example. And you may want to show how specific genes are expressed by those, in the context of those transcriptional states. So showing gene expression either as a UMAP or in other ways that I'm going to get to, is an important thing. You're going to want to accomplish all those visualization goals using good principles of data visualization. So I always recommend, and you'll see it done differently in papers, but always label all the axes and the facets and whatever dimension that you're trying to show, whether it's the size or the color or the shape of whatever aesthetic that you're plotting, always have it labeled, and have it labeled in a way that's clear and understandable to your reader. Okay, choosing the correct aesthetic for the correct type of data is important. Okay, so that, I think that's, we'll go through some examples of that. Choosing a color palette that is a good and even representation. The ones that are automatically built into the functions that I've written are chosen to be visually even. They don't highlight certain values inappropriately. They're usually colorblind safe, and these are all things that are important for your publication. And then you want to be aware of representing your data fairly. So, if you're plotting 100,000 cells and they're all very similar, they're all going to be stacked on top of each other. And that can cause some artifacts in terms of visual representation, and you have to be aware of that. You have to be aware that there's, because of the nature of this data, there's going to be many cells that don't express a particular gene. So it's called zero inflation. And you're going to have to keep that in mind for how you display your data. And then you will have to perhaps consider how your data is normalized and whether it's scaled between different genes, and there's different ways you can address that. So it's not entirely a straightforward choice in how to present your data. So how are you actually going to do it? Okay, so you're going to start always with a cell data set object as the core, if we're using this particular set of functions. Okay, we already discussed the reduced dimensions, and these are usually, and you can get these using this sort of command here to get the UMAP dimensions, and these are usually going to be mapped to the X and Y aesthetics. Okay, so that's the whole purpose, to show in two-dimensional space where your cell sits. Okay, so that's pretty straightforward. And then that whole table that we've been constructing with the clusters and with the sample metadata, all this can be plotted as a feature or as a dimension of your data, okay? And that's usually done as a color aesthetic, okay? Either a filled color or like the color of the outline of a color, okay? And those are usually discrete variables, but they can be continuous. So if you had, a certain value that you added that, was a number, for example, a continuous number, then that can also be plotted as a color scale. And the functions that I'll show you will be able to handle that. It's very easy, and I've shown this earlier, to identify your choices for what to plot by just looking at the cell metadata using these commands here, either the call data from the single cell experiment or BB cell meta from the BlazoTools package. Both of these are good for that. If you're plotting expression data, this is different from cell metadata. So this is expression based on the gene expression matrix at the center of these data objects. That's always going to be like a color scale, for the UMAP plots. You can get that using the expression command here, exprs from your CDS. That will give you the raw count matrix. These are, at least in the functions that we use, these are transformed on the fly using log transformation and size factor normalization. Now, the reason that is done is because you may want to change those factors. You have the ability to change how this transformation is done or how the size factor normalization is done. And so rather than store all those as separate matrix objects, which would be a very large data object, the computer does it sort of on the fly from the raw data. Okay, so that's basically how that's done. You may have other data, and we're not going to get into all this today or even in this particular course, but you may have other data that you want to store and visualize, and you can do like antibody binding from a site-seq experiment, or CRISPR enrichment from a CRISPR screen, or pseudotime if you're calculating pseudotime, and all these things can be plotted in a UMAP format. Okay. Now the plots that I highly recommend, and you will if you're using the functions that I'm showing you today. I recommend that you stick with ggplots. These are the most flexible and easiest to use format for plotting. They provide a consistent aesthetic, consistent palettes. They're easy to modify after they've been made from the base function. And so stick with that as much as you possibly can. We're not going to talk about heat maps. They're a little bit more complicated to get into. We might touch on those next week if we have some time. Okay. So let me demonstrate for you some examples of visualizations that I think are particularly effective and have been built into the BlazoTools package. Okay, so the first thing you might want to do is just plot your samples, okay, by in a UMAP plot. Okay, and so you can see here you've got the cells that we've analyzed, and you can see that the color indicates which sample that those cells came from. And the purpose of this data set, as a reminder, is to show that the Chromium controller and the Chromium X produce similar results. And as you can see, they generally do produce very similar results. All right. So you may want to plot clusters, okay? And you may want to, and I've already shown you how to plot clusters using the this command, this var UMAP command, but you can also highlight a value, okay? So let's say you wanted to highlight the T and NK cells. You can specify that, and you can change the cell size to make them a little bit bigger in this case. And so that highlights the T and NK cells. You can change the color of the cells, okay? So that palette argument, that will change the color for you, okay? You can, and this is a particularly useful function that I use, more or less all the time. You can plot the density of your cells in UMAP space. Okay, so you can see already, if I blow this up a little bit, you can see that there are some areas where there's a lot of cells stacked on top of each other, like here, and not so many cells, like here and here. And same thing, less cells there, more cells stacked on top of each other. So this is the overplotting problem that I've mentioned, okay? And you can be very quantitative about it and also visualize it using this command here. You can plot the density of the cells in those spaces. And that is a very effective way of showing that there's a lot of cells stacked up here, not so many there. And usually, and actually this particular command requires that you show two different, that you split it into two different facets. And so we're showing the Chromium machine here and the Chromium X machine here. Okay, and they look very similar. So that's good. So let's say you wanted to show expression of a particular gene to say, okay, I've called these T cells, but are they really T cells? And so that you would use this command, the BB gene UMAP, as opposed to the var or variable UMAP. You would use this gene UMAP, and you just simply specify which gene you want to plot. And something happened. Sorry about that. Try it again. Oh, what's going on? Oh, okay. So there you go. So there's a color scale showing expression of CD3 in these cells, which we've already identified as T cells and NK cells for the most part. You can plot multiple genes in a single plot. So that's how that's done. You just provide it as a vector. Okay. Another thing that is very powerful is you can, with this function, plot aggregated genes. In this case, I'm going to show you module 1. Okay. And remember, we calculated module 1 in the processing step. And we can generate a table of aggregated genes using this sort of command. And if you want to know exactly how that looks, all it is the gene ID here, the ensemble ID, together with the factor that we want to group by, in this case, the module labeled factor. Okay, and there's one line here for every row of genes that are in this particular module. Okay, so we run that, and then we can plot that, and there you go. So that's expression of module 1 genes here. Now, this is a really nice way to visualize groups of cells, and it counteracts the problem of zero inflation. So module 1 probably has, I don't know, 2,000 genes in it. So the likelihood of any particular cell not expressing any of those genes is very small. So every cell is going to be plotted here with a value, and that's helpful for the visualization. But you can still see that this highlights very strongly the monocytes and dendritic cells. So module 1 is a group of genes that are very specific to monocytes and dendritic cells. So that's another super useful way to plot your data. Okay, I think probably the most quantitative and the most information dense way to show expression of genes is with a gene dot plot. Okay, and in this particular plot, the size of a dot is going to indicate how many cells in a particular group, or in a particular group of cells, express a gene, and the color is going to tell you the level of expression for that gene. Okay, and you can see here, so expression is a color scale, proportion is a size scale. CD3 is very strongly expressed, both in terms of proportion, about 75%, and in level per cell expression, as you can see, it's in this bright yellow color. And that's for the T and K group, and it's not very highly expressed by the other cells. So it's a very specific marker. All right, so that gives you clustering, it gives you gene identification, and it gives you proportion of cells and the per cell expression. That's 4 dimensions of your data in one single plot. So that's a very information dense visualization that I really like a lot. The other thing you can do is you can reorder that. So the first way that I did that, it sort of automatically chose a clustering algorithm. Now it didn't show the dendrograms, but behind the scenes it was calculating the dendrograms for like how all those cell groups were related. But you can override that and just tell it what order to plot things in if you want to do that, and that's how you do that. You can even add a 5th dimension to your data. In this case, you can. It's a little bit more verbose, but you can do it, and this will split your plot into 2 or more facets in order to show like another variable. And in this case, I'll show you, we're going to map the equipment variable, in this case, either the Chromium machine or the Chromium X machine, to the facet dimension. And then we're going to leave basically everything else like it was. So if I run this, that will generate a table that's going to tell the dot plot function how to split up all the dimensions. And then I'm going to run this. And the key factor here is this group cells by multifactorial. That tells the command to use this particular table cell groupings to plot the data. Okay, so if you run it like that, then now you've got the equipment as yet another dimension of your data, and you can easily, rearrange or compare or however you want to do it. But you can look side by side and see, yeah, these are very similar data sets. The patterns look very similar. So that's a nice way to show that. Okay, so then the last visualization that I want to show is cluster representation. So an important concept for how you're going to analyze these data is what are the inherent differences between clusters of cells, number 1, and then number 2, what is the distribution of my cells between those clusters according to some experimental group. Okay, so let's say, for example, I had cluster A, cluster B, and I have my control group and my experimental group. If cluster A is 50-50 and cluster B is 50-50, then there's no difference between my control and experimental group. That's one situation. The other situation is maybe cluster A is 90-10 experimental and cluster B is 10-90 experimental. Well, that's like an important difference that we'd want to know. So you can get that from this particular command. Okay, so you plug in the CDS that you're using, you plug in the variable that you want to cluster by, okay, and you plug in the class that you want to use to ask what's the distribution of my class between these clusters, and you tell it which is experimental, which is control. That basically just tells you the direction that the plot's going to go, and you can run it like that. And you can see here that according to our clusters, the B cells are relatively enriched in the chromium sample. The DCs are also relatively enriched in the chromium sample, and the other ones are relatively enriched in the chromium X sample. Okay, and like none of these are particularly strikingly different. Okay, so these are not significant according to the Fisher exact test that we've used. This one is barely significant. And if you look at this, if you look at this density plot here, and we look at the B cells, which are, down here, you can see that the chromium, it's slightly, a little bit more, I don't know if you can even appreciate that on your screen, but that's a slightly lighter purple than the X cells. Okay, so there's a very slight difference, and it's definitely not something that I would consider maybe it's technically significant, but it's not like a compelling difference. But so that's how you would do that. And you can apply that to sort of any cluster that you want to generate in your data. Okay. All right. So the last thing I want to cover quickly is, since and we might run over a little bit here, and I apologize, but the last thing I want to cover is differential expression. Okay. And the important things to know about differential expression is that your goal is going to be to you have to decide how you want to do it. Do you want to get a list of all genes that are differentially expressed between two groups of cells, or do you want to get a list of, or do you want to like test specific genes and whether or not they are differentially expressed? And if you want to get like every gene, the best way to do that is with pseudo bulk analysis. And what this does is it takes groups of cells, it generates an aggregate count table, very similar, basically identical to what's used for traditional RNA-seq analysis, which you would normally run with DE-seq to analyze. This uses DE-seq to analyze this synthetic aggregated count table, and that basically does the same analysis that would have been done in a bulk data set, except for the fact that you can, as the programmer and analyzer and biologist, can decide which groups of cells you want to compare. So it gives you sort of infinitely more power than doing a sorted bulk RNA-seq experiment, but you get to use the same bioinformatic tools to do it, and you get the same sort of output that you would see, from sorting cells and then doing, bulk RNA-seq on those cells. So this is the recommended way to do it. The thing that I want to point out is that you need to have replicates, number one. Okay, so if you're running like a single sample, it's not really going to work well. Okay, let's say you're running one sample from each group. Okay, that's sort of like the minimum thing that you need. Okay, so a control and experimental, and you might even say, well, that's not so great. But remember, because you can group cells arbitrarily, you can take cells that are in a similar transcriptional state and use cells from sample 1 and sample 2 as replicates, and then use cells from sample 1 and sample 2 as replicates that are in a different transcriptional state, and use those as what I call pseudo-replicates. Okay, and so this basically, essentially doubles your number of samples that you can use to compare. Now, the caveat for this is what you're doing is you're not determining the differences between your samples. You're determining the differences between cell clusters. And this is the key thing, because it's a two-part analysis. You have to determine what's different between your cell clusters, and then what I just showed you with the bar graph, determine how your cells are distributed between those transcriptional states. And so this is the important concept that I want you to think about when you're doing this sort of analysis. So let me just point out a little bit more detail. Let's say you have, in this case, a very, high-end, well thought out experiment. You've got replicates, mouse 1, 2, and 3 in your control group, mouse 4, 5, and 6 are in your experimental group. Those are true biological replicates, maybe. Okay, so controlled and it's great. Problem is, there's only 3 per group. Okay, so maybe you want more. Maybe there's just like not enough cells from one of your samples or whatever. Then you can use what's called pseudo-replicates. Okay, and just to reinforce, a pseudo-replicate is a subset of cells from a biologically distinct sample. So say cell cluster 1 from mice 1 through 6 versus cell cluster 2 from mice 1 through 6. Okay, and so let me show you what I mean by this. So if I run this sample here, it's going to take the cell metadata from our CDS, okay, this big table, and it's going to group it not just by sample, but by sample and Leiden assignment, and it's going to summarize it. Okay, so I run that. This gives us my pseudo samples. Okay, so the 4 groups of cells from sample 1, which is the Chromium controller, and the 4 groups of cells from sample 2, which is from Chromium X. Okay, and so what I can do is I can use this. Let's say I want to determine the differences between TNNK and monocytes. So there's one replicate for TNNK, there's the other replicate for TNNK. Okay, hopefully that's pretty clear. And then all I do is I take that pseudo sample table and I plug it in to this command here, and that tells the computer that I want to consider these 8 pseudo samples, and I want to determine now the effect of Leiden assignment on gene expression. Okay, Leiden assignment is this value here. All right, and then I want to return the T and NK as a positive value. That's why it's here first, and monocytes as the reference or control value. Okay, so if I run this, and I already ran it, it's very quick, but I won't run it again. That will give you a list, okay? And the list has a lot of good information in it. All right, most important is the header, which tells you that the values are going to be, in this case, the mean of normalized count, let's see here. The standard error, for example, Leiden assignment of T and NK versus mono. So that confirms that T and NK is going to be positive. And if I run result, it's going to give you a table like this. And so it's just like the output that you get from DSeq. It gives you the gene name, it gives you the log fold change, it gives you p-values and adjusted p-values. So this is like what you can use for any sort of IPA analysis or gene set enrichment analysis or any other sort of data mining program that you want to use, either in R or anywhere else. And so this is telling you, again, differences between these 2 groups of cells. But you could have arbitrarily chosen different groups of cells, based on any other cell metadata factor that you had chosen. Okay, so that's pseudobulk RNA-seq. That's by and large the best way to go. There's another command which I use, very rarely these days, called, that involves a regression. That's better for, you can use that looking at individual genes. I will point out that in both cases, you can add the effect of confounding variables. So let's say batch or, whatever. Usually it's like some sort of a batch factor. You can add that in, just like this. Let's say plus batch. That will sort of regress away any effects related to batch and improve your detection of significant differences. Okay, so well, I think we mostly got caught up with everything we wanted to run through today. Sorry for running a little bit over. I know we went through a lot of this stuff pretty fast. There's hundreds of different variations and options that you can use for all of these commands. So you really just have to explore on your own what works best for you, your way of thinking about things, and your particular data. I would encourage you, you now have like basically all the tools to be able to analyze your own single cell RNA-seq data and get lots of good information about it. You just need to sit down and practice and do it. You can definitely start from the vignette CDS that I've provided in the Blazor data package that you can get, that you'll get basically with the Blazor tools. If you're able to install that, that will automatically come with it. Yeah, so use that if you don't have your own data yet, just to practice and to explore. Okay, thanks for sticking with me today, guys. Questions, put them in the issues section, or if you can't manage to find that, then just email them to me and I'll post it for anyone who's interested. All right, so we're going to wrap it up next week with a discussion of how to save all this data in a way that you can get at it again at any point in the future. All right, thanks a lot. Have a great week, everyone.